This is it, the last of my three-part series on the central dogma of molecular biology. Now let's look at translation, and this is going from mRNA to protein, which is our final molecule that we'd like to get. And there it is. Now, you may not recognize that right away, but I hope that upon closer inspection, you'll recognize that that's on the front cover of your textbook. And that right there is a ribosome. And if you look really closely at it, you can see this region right here, that's actually an embedded piece of nucleic acid. That right there is ribosomal RNA. And it is in complex with proteins in order to read mRNA and turn that into proteins. The coolest part, I think, is that the active site of this enzyme here, a ribosome, is actually made out of nucleic acid. This is probably one of the most primitive, but one of the most important enzymes in the entire cell. And of course, it's evolved to now be even more efficient by complexing with protein. So let's look at the overall picture one more time of what we're looking at in the central dogma. We have got mRNA. It's been processed and has left the nucleus. Now it's in the cytoplasm, and in the cytoplasm it's going to start to interact with a ribosome. And what that ribosome is going to do is it's going to take in another type of RNA called tRNA. These are transfer RNAs, each of which will be carrying a specific amino acid. And a 3-letter sequence on that tRNA is going to match with the 3-letter sequence on the mRNA to make sure that it delivers the correct amino acid in the correct order. And when it does, the ribosome will fuse those amino acids together and form a long chain of them, which ultimately becomes our protein. So here it is. This is one of the most incredible charts you will ever see. This is the language of DNA. We have learned to read it. And what you're looking at here is a chart that tells you what every 3-letter sequence in DNA means. So if the first letter is an A, and the second letter is a U, and the last letter is a G, we've got AUG written in the mRNA, which corresponds to the amino acid methionine, and that is the indicator for where to start making your protein. And any one of these 3-letter codes corresponds to another individual amino acid. And the absolute craziest bit of this is that every known species on Earth uses the identical sequence of 3-letter words to spell the exact same amino acids. This has been conserved throughout all of evolution. Imagine how devastating it would be to every protein in an organism's body if in the course of evolution suddenly a 3-letter word for methionine starts inserting threonine in the position where that amino acid should be. None of your proteins would work. That organism would surely die. So for the entire evolutionary history of all species on Earth since the dawn of time, this set of instructions has been preserved. And of course, the implication of that is massive in that we can take genes that are written in the instructions for any organism and move them from one organism to another. Your cells can read the instructions in a bacteria, and bacteria can read the instructions in yours. And this is the very root of genetic engineering, which is going to be our topic for next week. And so there's that molecule, the tRNA, that we're going to introduce into the mix. And essentially, that is a folded up piece of RNA. And on one end of that molecule is an amino acid. And on the other end of that molecule is a 3-letter sequence called the anticodon that is going to complementary base pair with our mRNA. So to translate the mRNA into protein, codons, which are these 3-letter words, must be read one at a time to assemble the amino acids in the right sequence. This occurs in the ribosome using tRNA. And so here's a little schematic of tRNA. When we break down how it folds, it's really got these 1, 2, 3 loops, and here's the end of the molecule. There's the 3' end, there's the 5' end, and this is the anticodon loop that is going to complementary base pair with a codon on the mRNA strand right there. And the key is that on the 3' end, okay, bound to it is an amino acid. So a tRNA molecule that has an amino acid attached, we're now going to call an aminoacyl tRNA. For short, we call them AA tRNAs. Okay, so this one is loaded up and ready to use. The anticodon is going to show it where it goes in sequence. So here's a little bit more of the chemistry of how it's bound. There's a little ester linkage to the 3' end. And here it's showing you the hydrogen bonding that's causing this molecule to fold. When we look at a more accurate picture of its three-dimensional shape, it looks something more like this. We can flesh it out with even more detail. Here you can see those loops color-coded with the nitrogenous bases sticking towards the middle, complementary base pairing with each other. And there you can see that free 3' end sticking out, ready to accept an amino acid. And so here's a really cool three-dimensional model of what a tRNA would actually look like if you could see it with your eyes. Amazing, isn't it? And so floating around in the cytoplasm is an enzyme for each of your different tRNAs. So the tRNAs are manufactured in the nucleolus and sent out to the cytoplasm, and waiting for them there are these enzymes that use a bit of ATP to bind the amino acid to them. So you can reset them after they get used, and you can load them as soon as they come out of the nucleus. And there's a different aminoacyl tRNA synthetase for every single tRNA that exists. So here's a nice little schematic that's a little more accurate than that animation you just saw of what these aminoacyl synthetases look like. There's a binding site right there for ATP, there's a binding site there for an amino acid, and there's a binding site right there for the tRNA. So we load it up, we place our tRNA inside, and now we have a what we call charged tRNA ready to use. Okay, so how many tRNAs are there? 4 nucleotides, or 4 different letters in our code, and we're making 3-letter words. How many different permutations of those 4 letters can you have? So how many 3-letter words can you make with 4 letters? And so if you know a bit of statistics, 4 choose 3 would give you 64 different combinations, but that should raise your eyebrows a little bit. 4 choose 3, 64, but aren't there only 20 different amino acids that we use? Yes, there are. There's a little challenge for you. You can pause the video and try to calculate how many combinations you could make if it was only a 2-letter word. And what you'll find is that you won't have enough if you have a 2-letter word, so it must be a 3-letter word, meaning that you're going to have extra combinations. And so if we look back to this codon chart, you can see that to insert leucine, there's actually 1, 2, 3, 4, 5, 6 different combinations of 3-letter words that all will tell it to insert the same amino acid. Okay, there's 4 different ones for proline, 4 different ones for threonine, 2 different ones for aspartic acid. And so you can see that there is what we call redundancy in this code. Okay, sometimes that even means that a mutation, let's say this C turned into an A, doesn't do anything at all to you. It doesn't change the order of amino acids in a protein. It's still going to code for proline. It's also interesting to note that not every single 3-letter word corresponds to a tRNA. There are 3 combinations of letters, and here they are, that we call stop codons. So if there are 64 total combinations, we only need 61 tRNAs to accomplish this codon chart in reality. Okay, 3 of them have no tRNAs associated with them. one of the reasons why this wobble might take effect, or why those particular letter combinations happen, and we can look back for a second at these, you can see usually the first two letters are the same, but it's only the third letter that changes, right? AC is going to give you threonine, GC is going to give you alanine, and so that third position is much less specific than the first two. We call this the wobble hypothesis. Which says that that third nucleotide in the anticodon is less important than the first two. And I think of it kind of like this little bozo punching bag. It's got that kind of wobble to it, and what's really fixing hard is one side of it, and the other side is curved up. So there's my little bozo the clown. Okay, so we say that that third nucleotide on the anticodon is in the wobble position. And so here you can see that shown visually, where you've got your mRNA sequence, and here you can see that whether it's a C or a U in here doesn't really matter, because this G isn't going to perfectly hydrogen bond with it anyways. It's on the curve of this anticodon loop, whereas these two are going to form perfect hydrogen bonds and make a strong link. Okay, in other words, both UCC and UCU are going to bring the amino acid serine. So what about this ribosome? So this thing is made out of two subunits, a small subunit and a large subunit, and they will only assemble when they bump into an mRNA. So the small ribosomal subunit will attach, and then the large ribosomal subunit will attach after that. And inside of the ribosome, there are three separate binding sites. There's the P site, which is going to hold one amino acyl tRNA, and it's going to be attached to the growing polypeptide chain. And then you have the A site, which is going to be able to accept and receive new incoming tRNAs. The last site is the E site, which is where tRNAs are going to exit or leave after they've been used. So let's have a look at this in action. So there's once again going to be a few steps to this. We've got initiation, elongation, and termination, and also once again we're going to have an extra step in eukaryotes for processing. So let's have a look at initiation. So the first thing that's going to occur is that the small ribosomal subunit is going to bump into an mRNA that's just left the nucleus, and as soon as it does, that's the trigger to help line up our first tRNA. And so here's how I'm going to represent that tRNA. There you can see the anticodon at the bottom. And the first one is always going to find the start codon, which is AUG. We'll revisit how that happens in a moment. And when those two assemble, that's the cue for the large ribosomal subunit to align itself so that the first amino acyl tRNA is in the P site. Now that this whole thing is assembled, we're ready to start. So there's initiation. You can pause the video and see if you can explain this to yourself or to a friend. Now let's have a look at elongation. So the next thing that's going to happen is that we're going to have another tRNA bind right beside it. And the A site is spaced so that it's exactly one codon over. So in the P site, we're bound to AUG, which means that this anticodon must read UAC. In the second location, AUC, we've got now U, A, and G. And so this different tRNA is carrying a different amino acid. So now we're in the A site, and once that thing is bound in the A site, the ribosome does its magic. Now it's not really magic, it facilitates a chemical reaction. Let's watch that again, where it's going to remove the amino acid from the tRNA in the P site and create a peptide bond between these two amino acids, right? That's our dehydration reaction. Bam! Now that's our cue for the entire ribosome to shift over on the mRNA molecule exactly the length of one codon, so 3 nucleotides over. Now you can see that the used tRNA is in the E site, and the tRNA that is attached to the growing polypeptide chain is in the P site, and look at that, the A site is free for us to repeat and do it again. So there goes our used tRNA, leaves from the exit site, and in comes a new amino acyl tRNA into the A site. And once again, react, shift, and exit. And this will happen over and over and over again until we reach a stop codon. So this growing polypeptide chain is going to start to worm its way out the top of our ribosome. And depending on the location of that ribosome, it can make it into different places. Maybe it's going to start putting it into the endoplasmic reticulum. Maybe it's just going to make it free-floating in the cytoplasm. And so once again, this is a nice summary video where you can pause and see if you can explain what's happening in this video to yourself or to a friend. So we're at the end, termination, where we're going to figure out where to stop translating that mRNA into protein. So there he is, the Terminator, and boy does he ever love his proteins. So here we are, and you can see we've made a large polypeptide, and we're coming close to the end of our molecule. I'd also like to point out that once again we're moving in the 5' to 3' direction, okay, along this mRNA. So that's been an ongoing theme throughout the entire central dogma. All of these enzymes seem to work coincidentally in the same direction, always 5' to 3'. And so here we have in the A site the codon UAG. And if you double check that codon chart, that is a stop codon. And there is no tRNA that is going to bind there. And so translation has stopped. What does bind instead is a protein, and it's called a release factor. And as soon as that binds, it causes the entire setup to break apart and causes a chemical reaction that will release the polypeptide chain. And so there they go, and we're left with our mRNA and our protein, and that ribosome is ready to be reused. The small ribosomal subunit may very well reattach to the beginning of this molecule and start again and make another copy. So here it is, the release factor, doing its work for termination. Pause the video and see if you can explain to yourself or to a friend what's going on. So here's an animation where you can see all of it happening in what looks kind of like real time, with a few additional factors that I didn't mention. So here you can see these dark blue tRNAs entering the A site, and here you can see with the help of another protein, it's pushing it along one codon at a time, and every time these guys go into the A site, it's linking to the growing polypeptide chain, and out through the E site, the used tRNAs go. Okay, and so eventually you can see that this growing polypeptide starts to emerge out of the ribosome. And here's a really cool protein that has bound to it and fused it with the membrane of the endoplasmic reticulum. And look what happens now. Okay, this is why the rough endoplasmic reticulum is rough. Very many of the active ribosomes are physically attached to it. Okay, you have this little protein right here, and you can see that the protein is now being made directly through a pore. And instead of coming out in the cytoplasm, this protein is now being synthesized directly into the lumen of the ER. And that's going to be really important for our next step in both determining what that protein is going to look like, how it works, and where it ends up. So before we get to that step, let's just have a look at this. Just like we could have multiple RNA polymerases working on the same strand of DNA to make multiple mRNAs, we can do the same thing with ribosomes. Each one of these represents a different ribosome bound to the same mRNA. 1, 2, 3, 4, 5 of them in a row. And as they're moving farther along, you can see the protein has gotten longer and longer and longer. Okay, we call this a polysome, or your textbook calls it a polyribosome. And so this is a way that we can make a lot of protein really fast. So now we've got tons and tons of mRNAs interacting with lots of ribosomes over and over again to make thousands and thousands of copies of protein really fast. This is why, when you go outside in a cold winter's day, within seconds your nose can start to produce huge amounts of the protein mucus to help slow down that air and warm it up before it hits your lungs. Okay, let's have a look at this under the electron microscope. So here you can see an mRNA with many ribosomes bound. So we would call this a polysome, and out from the top you can see the proteins emerging. And since the proteins look really long over here, I would assume that this end is the beginning of that mRNA sequence. That's the 5' end. Okay, and here it is again. But this now is in a prokaryote. And if you look in that prokaryote, we've got our DNA molecule there. Our RNAs are being produced all along it, and immediately once those RNAs are on there, the polysome attaches on and starts turning it immediately into protein. Okay, so let's not forget that what determines the function of a protein is the 3D shape. And all we've made is a skinny long chain. What's crazy important is that we need to fold this protein into the correct and functional final shape. And this is why in eukaryotic cells in particular, most of these proteins get made directly into another organelle, usually into the endoplasmic reticulum, where there are already some proteins waiting to help fold them. Sometimes we call these chaperone proteins that help to fold them up. Like this beautiful folded up piece of paper that looks like Yoda, you can see it's actually made out of multiple different pieces of paper, right? This has quaternary structure, and so we can also assemble multiple subunits. And what's really cool is that one single polypeptide chain, depending on how you fold it and cut it and modify it and assemble it with other parts, can do many different jobs. So once again, now one gene can make multiple RNAs in the same family. And then each one of those RNAs can produce a protein that gets folded or cut or modified to make a whole family then of related proteins. So a single gene doesn't actually make just a single protein. One set of instructions can actually make a tremendous variety of different and functional proteins for all kinds of jobs. And that's why it doesn't take a huge number of instructions to make a human being, but we have an incredible amount of complexity and way more proteins than we have instructions to make proteins specifically in our DNA. So we call this last step post-translational modifications, and that's fancy words for saying what sorts of things can we change about a protein after it's already been made or translated. So we can remove some amino acids from it. We can divide it into smaller pieces, right? Chop it up. We can add sugar or phosphate to it, right? Make it a glycoprotein or phosphorylate it. Or we can add several polypeptides together to give it quaternary structure. And so there's the central dogma, guys. DNA to RNA, RNA to protein. And I challenge you to pause the video here and see if you can work your way through a complete explanation of what's happening at every step of the entire central dogma, all the way through to fully folded and functional protein. This final diagram is probably our most complex, that includes every detail that we've talked about through the entire series of 3 video, or for the transcription and translation video. So once again, pause it and see if you can explain every step that you see. So the last step is to follow the link in the description to check out a beautiful 3D animation from the HHMI animations that'll work you through the details of translation as well. Enjoy!
Work hard and stay safe.